Hello everyone, my name is Kaitlyn Charmley and I'm one of Dr. Kathleen Robert's master's students based out of the Marine Institute at the Memorial University of Newfoundland and Labrador. This presentation is going to be on my work analyzing the seasonal change between benthic habitats in a subarctic ecosystem, which is part of a larger interannual monitoring project funded by MEOPAR, which attempts to model species-environment relationships across both space and time in the subarctic benthic communities of Conception Bay, Newfoundland. The health of marine ecosystems on which we and all other species depend on is deteriorating at an unprecedented rate due to climate change and anthropogenic exploitations. To name a few, there's habitat degradation, ghost fishing from lost fishing gear, and drastic species decline like we have seen personally here in Newfoundland, with the loss of cod having a huge impact on communities. Benthic marine environments are no stranger to this deterioration and are exploited every single day around the world. To manage these areas, ecosystem baselines must be properly characterized and understood so that changes outside of normal fluctuations can be detected and monitored. Many researchers do this by surveying the area once to collect ground truthing data and creating a habitat map of the ecosystem at that one moment in time, which makes this map a simple snapshot of the ecosystem and doesn't incorporate temporal variability. But with the knowledge that ecosystems are not static, how can we be sure that any shift from that snapshot baseline is representative of actual changing ecosystem and not just normal seasonal fluctuations? Atlantic Canada in particular is one region that is predicted to be highly threatened by global climate change. Just west of St. John's in Newfoundland and Labrador, I carried out a seasonal repetition of ground truthing in Holyrood, Conception Bay, a 23-kilometer square bay with a subarctic climate. To quantify the level of spatio-temporal changes in a benthic community by comparing the production of habitat maps across the seasons. To do that, sites were chosen using previously collected data to capture a variety of depths and seafloor types. And then beginning in summer 2020 and repeated in the subsequent fall, winter, and spring, we collected video and environmental data at 68 sample sites across 3 days using a tow camera seen here on the right. So next, we clustered the sites by their species assemblages and then predicted the different assemblages back onto the map. So the species seen below are that cluster's dominant and identifying species. When comparing the clusters by season, the assemblages found in each season changed drastically by presence and by size. Notably, we saw clusters combine into larger clusters, so the red cluster, which was dominated by stars and sand dollars, and the orange cluster, which was dominated by the green sea urchins, combined into one larger cluster in the fall and then separated back out in the winter and spring. And also the blue cluster, which was dominated by a type of sea cucumber, was commonly found in the summer, completely disappeared in the fall, was very minimal in the winter, and then returned in the spring in very large numbers. But when it returned this time, it mixed with the yellow cluster, which represented brittle stars, as they were commonly found together in this season. One of the really interesting stories here was the disappearance and reappearance of the blue cluster, which was dominated by a Solus sea cucumber. These photos were taken on the same transect, and as you can see, there was a drastic decline in this species in the fall and the winter. In the literature, not much is known about the life cycle of this species of sea cucumber. It's possible this species is burrowing into the sediment or maybe migrating. Other species of sea cucumber have been documented migrating seasonally, particularly one study here in Newfoundland on a different species of sea cucumber documented a sudden movement at sexual maturity from shallow to deep water during the fall when temperatures decreased rapidly. This area we are studying only goes to a maximum of 100 meters, so it's possible that we are solely observing the shallower range of this species niche. Another example of seasonal movement that we saw here in Holyrood was snow crab, which are an economically important species here in Newfoundland. We observed them vary in location that they were found throughout the seasons. We found that their movement is mainly controlled by temperature and depth. Notably, it appears that snow crabs are likely moving to warmer, shallower water in the spring, which coincides with previous literature as well. And those are just two examples of the movement that we observed occur throughout the seasons. So the results from this project support the idea that one ground truthing event in creating habitat maps for management purposes is not enough, and can miss entire species or misplace the location of species. If at all possible, we really do recommend researchers or surveyors collect their data at more than one time in the year. However, if this is not possible, then we suggest researchers choose the season they study based on what sort of information they're trying to study. So for example, if they're just interested in the diversity of species that can live in an area, maybe they should choose to study in the spring when we found the most transient species. And on the other hand, if they're looking at an average representation of what species normally live in an area, maybe they should avoid the spring, where other patterns and assemblages could be masked by massive surges of temporary species. Also, if using this map as a baseline to compare future years against, the same season must be studied as was used to make the baseline, and cannot be extrapolated to explain the entire year. Thank you so much for the opportunity to speak today. If you would like to learn more about this project, you can go to our lab website, www.4d-oceans.com, or follow our lab on Twitter at 4DOceansLab. And my email is also right here in the bottom right if you would like to contact me after this presentation. Thanks again.